Good afternoon, and welcome to our MDICx webinar. I'm Carolyn Hiller. I'm Program Director for the Clinical Diagnostics Program here at MDIC. I'd like to introduce our panelists to you. We have JD Alvarez, who is the VP of Oncology Diagnostics at Janssen Oncology at Janssen R&D. JD is the chair of the SRS project. We also have Litai Feng, who is a staff scientist with bioinformatics for Roche Sequencing Solutions. Litai leads our data integration aspect of the project. Alex Lovejoy, who is the principal scientist for Roche Sequencing Solutions, has led our hotspot variants initiative. We have Jason Merker, who is the Associate Professor of Pathology and Laboratory Medicine and Genetics at the UNC School of Medicine and Lineberger Comprehensive Cancer Center. He chairs our SRS data validation component of the project. We have Julie Schneider, who's the Acting Associate Director for Research Strategy and Partnerships for Oncology Center of Excellence at CDER. Julie co-chairs our RFI and RFP process. Zivana Tezak is the Associate Director for Science and Technology of Personalized Medicine at OIR FDA. She is our primary contact for the project with CDRH. We also have Barbara Zehnbauer. She's the Steering Committee Chair for SpotDX Quality Assurance Pilot Project. Barb co-chairs the SRS, RFI, and RFP aspect of the project with Julie. Barb also led our landscape analysis initiative. Now I'm going to provide just a high-level overview of MDIC for those that aren't familiar with MDIC. MDIC is a 501c3 nonprofit organization and was organized to advance regulatory science of medical devices for patient benefit. We focus on science, not on policy. Within MDIC, we have a robust portfolio of programs that we focus on. This project lies within the Clinical Diagnostic Project. If you'd like to learn more about the MDIC projects and programs, please visit our website, which is www.mdic.org. And now I'm going to turn it over to our project lead, JD Alvarez.
Thank you, Carolyn. So as Carolyn said, I'm JD Alvarez. I have the pleasure of chairing this program that has been going on for a little bit more than a year now. And the goal of this project is to develop reference samples that will be publicly available and that can improve the accuracy and reliability and transparency of NGS-based oncology tests. It's a collaborative effort to develop these reference samples, and we believe that when we can develop these, they will aid in efficient NGS test development and validation, and streamline and hopefully obviate some steps in the regulatory review process for approval of diagnostic tests. Additionally, transparency will be available, and this will compress development timelines for development of therapeutics. So next slide, please. As I said, we kicked off maybe a little more than a year ago, and we've divided this project into two phases. The first phase is scoping, where we were able to publish a detailed description of a prioritized set of samples and variants that need to be characterized and made available. And as we're doing now, we're planning for the development and characterization so we can move into phase 2, which is to pilot and characterize and scale up, essentially to make the product. Under that, we would like to contract the production and characterization of the samples that contain the high priority oncology NGS variants. And we want to transfer those outputs to interested commercial entities, which is why we're having this, this webinar. So ideally that these products will be made widely available and to the public, and will be finished within 1 to 1.5 years. And I will now turn it over to Barb Zehnbauer to go through the RFP and the timeline.
Thank you, JD. Hopefully all of the people who are participating here have already had a chance to look at the RFP that was posted at the MDIC website. You should be looking at a slide now that says RFP time period. This is telling you what the deadline is. It was originally posted on August 29th, and we will be accepting proposals at the website through 5 p.m. Monday, September 30th, so the end of this month. And it can be downloaded from the link you see here. Next slide, please. The format is just a Word document. We would like it to be blinded so that the reviewers are not informed about the entity that is submitting the proposal. We'd like it to be as fair and unbiased as possible. I realize that some of the participants today have already participated in a previous RFI webinar we had. We thank you very much for your input there. It did help guide our proposal design, our RFP design. So you see the elements here that we're going to request for people to provide in their proposal. We have not specified a page limit, but we would encourage everyone to be concise. It really facilitates the review process. And we would request that an itemized budget be provided in a spreadsheet format. I'll talk a little bit more about that. You see the email address here for submitting the completed proposals. Next slide, please. So I'm not going to read through the RFP, obviously. In time limitation, we want to preserve this time for your questions to clarify issues that are already in the RFP. Next slide, but I will provide just a brief overview of the contents of the request for proposals document. Most of the description centers around, as you might expect, the technical approach. And we had a committee of people that designed this RFP. We went back and forth on a lot of these details, and our main purpose here today is answer your questions about things that may still not be quite clear or you'd like to have better definition. So you'll see in the document that you've hopefully had a chance to review that we spent a fair bit of time talking about we want very good descriptions of the parent cell line that's going to be selected and how well characterized it is genetically. We are asking for human cell lines to be engineered with the variants that have been identified by our SRS group as the priority variants, primarily variants that are not currently available from commercial entities, but would help facilitate new assay development by biotech and pharma companies. We have specified that we want these variants introduced into the endogenous position in the genome to facilitate analysis. We also envision that these will be useful in constructing cell line mixtures with 10% variant allele fractions for each of the priority variants. So the parent cell line plus the derived engineered cell lines would be mixed. For our validation purposes at MDIC, we will request, in addition, genomic DNA from these components to be verified prior to the final stage, which we envision will be those same cell lines mixtures grown up, pelleted, formalin fixed, and paraffin embedded, and those would be the final product that new assay developers say they are most interested in acquiring. These are the items and types of samples that most closely replicate the actual clinical samples that their assays are going to be evaluating in practice. We also have several sections in the RFP defining the quality considerations that we want the proposals to include, the validation data that's coming from the manufacturer, in addition to the validation that will be generated by our MDIC laboratories. And finally, we do want a specific timeline to be described for each of these both final deliverables and intermediate deliverables, and a budget that is itemized for each of those types of steps. This might be a little more clear in the next slide. So we broke it down into the different deliverables that you'll see described in the RFP, parental cell lines, the engineered variant cell lines, and making mixtures of those two are the first level or primary products that will be manufactured. But those are not what we envision to be the final product that will be utilized by assay developers. Then from each of those components, we would expect genomic DNA to be produced, and those genomic DNA preps representing both the parent cell line, each derived cell line, and the mixtures would be distributed to MDIC laboratories for validation. This is sort of a proof of concept step, if you will, verifying that what we ask to have designed is actually performing the way we envision it to be performing in next generation sequencing. We recognize that not all vendors necessarily do an internal validation with next generation sequencing. Many times it's other types of molecular pathology assays that we use. Then the final product that we would say would be commercialized and would be distributed more widely to assay developers is the final one you see, the FFPE cell pellets of the mixtures of the variant cell lines and the parental cell lines. And I'll go ahead and go to the next slide, please. We have a description at the end of the RFP to let everyone know what kind of evaluation criteria we're going to be looking at. Obviously, the technical approach that you're going to describe and how which cell lines are selected, how the variants are engineered, how they are validated is going to be key. One of the largest parts of the evaluation measurement, if you will. We also want to hear discrete descriptions of the organizational experience that each submitter has with regards to both developing reference samples of this type, what their outline and practices are for quality systems management, and how the overall project will be managed, adhering to the timeline that you described, on how we will have validation and verification of each milestone that's being met. And then a description of what the facilities and equipment capabilities are for the manufacture and production of the kind of reference samples that we're describing in the RFP. With that, I think I can turn it on over to Carolyn.
Great, thank you, Barb. I know some of you may have some questions on the business component and the legalese that's included in the RFP. And I have with me in the room, I have Lenwood Falcon, who is MDIC's General Counsel, and will be able to answer any questions that you do have. I forgot to mention early on, for the question and answer portion, if you could enter in your questions into either the chat box or the Q&A, we'll be able to work through those after our discussion here. But one of the things that we want to bring out is MDIC would like to develop a long-term partnership with the vendors that we select to move ahead and engineer the samples. And so we do not intend to take physical possession of the validated samples, but we must control the access to the samples, as this is a collaborative effort. And I know our stakeholders and those within the research community are counting on being able to have access to the different samples once they're created and validated. We do not require vendor IP, nor do we need access to it. We don't want the samples to be handcuffed by vendor IP. In addition, we will be licensing them. Again, we're doing this to maintain broad access. The licensing, not that any of the vendors would do this, but we want to make sure that we have safeguards in place so that we do not have any price gouging for the end product that we have validated and to be used in test development and submission processes. The vendors, we're anticipating that we would like for the vendors to be able to make a fair profit. There is going to be ongoing expenses with, for lack of better words, the care and feeding of these samples so that they remain long-term available to the research community. We will be taking a percentage of the revenues to cover ongoing administrative expenses that we anticipate with the samples. We will be considering multiple vendors to ensure continued access. And one thing that we hope does not happen, but if one of the vendors that had the final product was to run into financial hardship or it looked like the business was going to close, MDIC would want to be able to move the samples at that time to another vendor so that the samples could remain accessible to the research community. So I hope that answers some of the questions that some of you may have. And I'm now going to open it up for questions. If anybody has questions that you would like, again, please type them into the question box and we will read them off. So, I'm seeing no questions yet. Oh, wait a minute. Okay, the question is, and I will read the question off, and JD, our project chair, will either answer it or pass the question off to one of our panelists. Question that we have, how many total cell lines are to be created?
Yeah, so I guess I'll pass that off to Barb.
Yes, we haven't specified a discrete number of cell lines, and it's one of the topics that we would like the submitters to describe at length. So for example, one could envision one cell line for each variant that's described. That would be a lot of cell lines and rather burdensome for companies to implement in new assay development. So we recognize that some of the priority variants could be combined in a single cell line that's derived from the parent, but perhaps not all 60 variants can be accommodated in a single cell line. We decided to leave the combinations up to the discretion of the experts who design these reference samples, that is, the people that are submitting these proposals. But we will expect to have specific descriptions of how you are going to combine variants in an engineered cell line, the rationale for doing so, how many resulting cell lines there are, and what the combinations of variants are in each of those derived cell lines. We recognize that different combinations will have different growth characteristics, and you probably have much more experience with that, and we didn't want to overstep that expertise. We'd like to hear your justifications for the combinations that you will propose. Thank you.
Next question. Is there any funding support for vendors' development and production work? I'll take this, JD. Yes. The RFP, we're looking in the proposal for how much the vendors anticipate that it will cost to make these samples, and MDIC, through collective fundraising with our stakeholders, will then be paying for that. So MDIC is and our stakeholders are putting skin into the game for the end product. Next question. On the RFI, it was specified that you do not want genome in a bottle samples, but the RFP doesn't state this emphatically. I assume there will only be one genetic background if we are pooling as well. JD?
Yes, I would, again, pass that one to Barb.
Yeah, that is correct. We would like all of the engineered cell lines containing the variants that are prioritized to be in the same parental background. So there's one parent cell line. We wanted a parent cell line that was relatively well characterized for other positions in the genome. And we look forward to reviewing the justifications for whatever cell line a proposal is presenting. What's the justifications for selecting the one that you're using? Genome in a bottle is useful for some combinations. We're not limiting people to using genome in a bottle, but we want to hear discrete justifications for the selection that you do propose to use. All of the variant-derived and engineered cell lines should be in that same parental background.
No, this is Zivana. I just want to add to it too that ideally why we said in RFI that genome in the bottle may not be the best background is because it's been extensively used, but it's a germline background. So ideally, if there's some kind of cancer cell line background that can be used, that this project can contribute, that would be our preference. But we understand that genome in the bottle is the best characterized out there. So, we'll see what we get. But we would prefer cancer cell line background if possible.
Zivana, this is Carolyn. Is there any differences with the genome in a bottle samples, as they do have an assortment of them available?
Yes, they have a number of samples. So ideally, it would not be the NA12878 because that's been so extensively used and everybody's been using it in the assay development. But they have several trios too that are well characterized. But
great, thank you.
The only thing is they're all germline and all
okay.
Normal samples.
Okay, thank you. Another question, do all the variants on the list need to be ready on day one? RFP states preference for CRISPR, but are you also open to other methods such as synthetic spike-ins?
So I'll answer the first question about the variants beyond this being needed ready for day one. I would say not necessarily. I would say we would like to see what people can propose, and if that means that their proposal comes with a staggered release, that could be considered. Maybe Barb or Julie or Zivana might want to weigh in. In terms of the preference for CRISPR, I will leave that to Barb or Jason or Lee Tai.
No, JD's absolutely right. Yeah, if we are open to a staggered production timeline, assuming that there is good justification presented for that in the proposal. With regards to spike in synthetic constructs, we specifically prefer that the variants are integrated into the host genome and integrated at the endogenous gene location. If there is some technical reason why a particular variant cannot be engineered that way, a very strong justification should be provided, but it is not the preferred design for this whole venture to have synthetic variant sequences spiked into a parent cell line genome.
Okay, next question. Does the estimated 1 to 1.5 years of phase 2 include PPFE sample production or calculating based on the gDNA submission to MDIC?
So I would say the 1 to 1.5 years is an estimate. Certainly we'd like to get the FFPE sample production as soon as possible. We'll have to see what what timelines are proposed. In my thinking, but others want to weigh in.
Yeah, so this is Barb. I think one of the concerns, and you'll see it described in the RFP, is that we want to make sure that there's not, I guess, genetic drift or further genetic changes in whatever original material is produced, the original cell line. So we'd like to see the cell line engineering construction described in one part of the timeline, and then the cells that are grown up from that batch, both for the genomic DNA isolation as well as for the FFPE sample production, should be all from the same batch, so that we know that we are verifying or validating the material that's actually going to be finally distributed. I don't know what exactly the size of that should be. That's something that we'll have to see described in the proposals. And I would assume that the vendors who manufacture these reference cell lines have experience in estimating this. Julie, do you have anything to add?
No, Barb, I think that sums it up.
Okay, next question. Does the engineering have to be scarless?
Yes.
Yeah, sorry, I was on mute. So, yes.
Okay, next question. Why will there only be a publicly available FFPE product? Why not distribute cells or DNA?
I think that's mostly a business decision on the part of the vendors. If you have a market for the other products that you've created, then there's nothing to restrict you from marketing that. It's just that when we had our initial conversations and input from the biotechnology companies that are developing assays for personalized medicine, what they specified is that FFPE human cell lines were going to be the most useful for them in terms of gauging how their products or how their assays were going to perform in actual clinical practice. Granted that a cell line FFPE does not represent the total complexity of a clinical tissue sample that appears in a pathology laboratory, but rather than cell lines or isolated DNA, these companies were saying that an FFPE cell line was their preferred product for using. You're right that other markets may make use of those intermediate products, and that's fine. That's just not the final product that MDIC SRS is specifying right now, but that certainly would be a business decision up to the individual vendor.
Thanks, Barb. Also, on the finished product, as far as the distribution, we anticipate that the finished samples will remain in which the vendors that we select in their product catalog. We will be branding them as the MDIC SRS samples, whatever the name is, so that individuals know that they are using the specific samples that have been validated by the MDIC community for use. But those will stay with the different vendors. Another question that I had received earlier was page limit. There's no page limit that's listed in the RFP. However, we encourage you to be concise and to tell your story and what you can do, and use the length that you need to tell that story. If your proposal comes with an excessive amount of paper and pages, I anticipate that will not be viewed favorably by the reviewers of the different proposals. So use what you need to tell what you can do in the best way you can. Here's another question. Is there any suggestions on how many copies of gene to be engineered into the cell line in the amplification type of variants? In the clinical setting, the gene amplification often seen could be over 10 copies or even over 100 copies, which would be very difficult to do engineering.
Yeah, so I will turn that over to Barb too.
I would turn that over to Lee Tai or Jason, because those, I mean, are someone who's developed the priority variant list. That was not part of the RFP development. Anybody?
I was not in.
Yeah.
I was not involved in those conversations either, but I think a reasonable estimate would be in the at least 10-fold amplification range.
Yeah.
I would agree with that. I think the idea is usually this can always dilute it and get smaller amplifications, but can't go the other way around. So I'd lean on the high side, and I'd say at least 10-fold is pretty reasonable. I think in general, users are going to want to get down to 5-fold or less with those dilutions, but I think 10-fold is a good target.
Okay, another question is, okay, I'm catching some feedback. Perhaps we can mute. Okay. As you mentioned, the pricing limit of off-the-shelf product will be determined by the license. Can manufacturers market and sell intermediates developed during this project? That's a good question. However, I think what everybody is looking forward to is the validated samples. And so until we know that the mutations are engineered into the samples where we said they were going to be, I don't think that the products will be ready for distribution. JD, others?
I would say it's possible that they would be useful. I'm not thinking of things off the top of my head, but I would defer to MDIC in terms of, you know, if the manufacturer thought that they had a market for those intermediates, would that be okay for them to market them? I would defer to them.
Yeah, this is Barb. I agree with Carolyn that, you know, the whole point of this is to have an MDIC validation that whatever is manufactured fits the design that we requested. And until that step's completed, it would be of benefit to hold it there. But as I answered previously, marketing those intermediate products subsequent to the MDIC validation could be envisioned.
And I think the details of that will be worked out with the contracting with the selected vendors. Another question. For the request about the material must be made from the same batch, is the same cell line bank sufficient?
We'll kick that to Barb.
I don't think I understand the question.
Okay, let me read it again. I might have messed it up. For the request about the material must be made from the same batch,
right?
Is the same cell line bank sufficient?
That's a question.
As long as there is a well-described quality assurance step that there has been no additional genetic variation occurring in the cell line, I think that would be acceptable.
Okay. Next question. Can manufacturers market and sell intermediates developed when making the final product? I think we just answered that from someone else. Next question. Do the amplifications have to be the whole gene, or would regions be sufficient?
I will kick that to Jason or Lee Tai.
So for the amplification region, in the variant list, the region is listed, so it just has to be that region.
No, I think it's the whole gene, isn't it, for the amplifications? Am I mistaken?
Yeah, so it depends on what's on the list. If it lists a region, then it would be the region. If it says a gene, then it would be the gene.
So yeah, if it's listed as the whole gene, it should be the whole gene. It should include the whole gene.
Okay, thank you. Another question about the pricing limit question. We can work this out, but again, what we want is we want a fair relationship. We're looking for samples that would be accessible. We want for them to be able to be used by the largest companies, as well as a very innovative researcher that may be at a small academic institution with limited budget. So we will get into those further in the contracting phase, but again, the anticipation is in our long-term relationship, we do expect for the vendors to be able to make a fair profit off of the samples that they sell. Okay, do we have any other questions?
I think you have the validation. What does validation data include? And I think
absolutely. Good one. Thanks, Zivana. What does the validation include?
And I will leave that to Barb and Julie. Sounds like we may have lost Barb.
Oh, sorry, I'm here. Would you repeat the question, please?
Weigh in too. Sorry.
What does the validation data include?
So we have a data validation group that is designing that. Basically, the genomic DNA from each of the 3 different preps will be subjected to next generation sequencing by a number of different labs. I'm not sure how many different labs. That's up to that group to decide. And then they will analyze the data and verify that the described variants are inserted at the proper location and they perform appropriately in the assay. I mean, Lee Tai, do you want to add anything to that?
I think that is it, yeah. Sequencing data to make sure that the variant engineer is there, and also to make sure that there is no additional changes that that we didn't expect.
And this is Julie. I was just going to add that on page 5 of the RFP under point 3, there is a list of different validation methods that might be used by the vendor to confirm. So, digital PCR, Sanger sequencing, targeted NGS are all listed there. So there is a little bit more information available in the RFP.
And that goes back to this, Zivana, to the question, what is exactly the question, the validation by the RFP applicant or validation by the MDIC working group that's working on validation, because those are 2 parts.
Right. So we're requesting, correct me if I'm wrong, Barb, this is Julie, we're requesting information from the vendor that they have in terms of their validation, but then there will be additional validation that's done by the MDIC data integration and data validation groups.
That's correct. So I mean, with all of these products, the primary ones and the intermediate ones, we expect that the vendors will describe their internal validation processes and assays. But then there are 3 steps where genomic DNA will be sent to MDIC participating laboratories for verification in a proof of concept step. And those will be validated with next generation sequencing assays.
I want to build on this. After we have validated it, we do plan on taking the data that is generated, and we will be posting that on, we've identified Precision FDA and site at NIH, so that data is publicly available to be used by researchers and to begin developing a public database.
So we are trying to learn and follow the genome in the bottle example as much as possible.
Okay, are there any other questions from individuals on the line?
You got another question.
Oh, okay. Some of the engineer's edits may be very challenging. If failures occur, this may be unavoidable. The vendor can try multiple attempts, but not everything may be possible.
I think that's a good comment. You know, certainly we're going to try to get as much as we can, but you know, obviously, if someone can demonstrate that they're unable to get this done for a specific variant, then you know we'll have to live with that.
And this is Julie. I was just going to add, if you have a sense of which ones might be more challenging, you know, providing information about that in the RFP response, I think would be helpful as well.
That certainly is true, Julie. The other aspect that we haven't ruled out is that some vendors or some methods may be better suited to some of these variants than others. We have not ruled out the option of having more than one vendor being awarded funds to make the spectrum of variants they can describe.
Okay, I do not see any additional questions. Panelists, is there anything that you would like to say before we wrap it up?
Thank everyone for their participation and really great questions.
Yeah, I'll echo that. I want to express our appreciation for everyone taking the time, likely at lunchtime, to talk about this. We're looking forward to seeing a number of robust proposals and moving this project forward.
I have one question. This is Barb. If any of the participants have additional questions after the webinar, will they have an opportunity to submit them prior to submitting their proposal?
Sure, you can send them to the email address that we have listed here. That is, wait, that's not, I don't have it. SRS-RFP@mdic.org.
It looks like you have one more question about when the recording will be available.
Okay. The recording will be available as soon as our communications department can do their magic and we'll have it posted online. I will send an email to the individuals who registered for the webinar. I will include a PDF of the slides that we showed today, as well as a link to where the webinar will be found. It will be on our website under MDICx, a panel that's in, I think, the banner ribbon, and we have an archived section. It will be in the archives, and we'll also put it on the SRS web project web page that we have, so that will be a ready link. Okay, I think we have everything, and again, thank you to everybody that's on the line, to the volunteers who have worked collaboratively to bring the project along. It's very exciting to look at this community effort to bring these needed reference samples to help patients. And I thank all of you for helping us with this and wish you a very good day. Goodbye.